More than any other organ, the brain is sealed off from the outside world and the rest of the body. There are many layers of tissue, bone, and skin to keep it protected from the outside, but there's also a blood-brain barrier that keeps out many substances circulating in the blood. Oxygen and nutrients can cross the blood-brain barrier, but many dangerous substances like bacteria cannot. With his skull fractured, Phineas's exposed brain is wide open, making him an ideal candidate for a fatal infection. No one in Cavendish in 1848, no scientist in America or Europe, has the slightest notion that bacteria can cause infection. Medical science in 1848 knows very little about bacteria, even though they were first seen through microscopes nearly 200 years before. Today we are used to seeing the microscopic world, but when the microscope was invented in the middle of the 17th century, it caused a sensation. The microscope became a new kind of high-tech entertainment for cultured gentlemen, and in 1655 an Englishman named Robert Hooke came up with a microscopic hit. He showed off a slide he'd made of extremely thin slice of cork. Under the microscope lens, Hooke saw that the tissue inside a cork tree was made up of rows of tiny box-like structures. They reminded him of the bare rooms used by monks in a monastery. Hooke called them cells. His cork cells, though, were empty because they were dead and dried out. It would take 2 centuries to figure out that it's the living stuff inside cells that makes them the fundamental unit of life. While Hooke was showing off his cells, a sharp-eyed Dutch merchant named Anton van Leeuwenhoek was making more powerful microscopes. Leeuwenhoek took a single drop of water from a rain barrel and turned his microscope on it. In that drop of water, Leeuwenhoek found a new planet of very, very small life forms. Animalcules, he called them. Leeuwenhoek was the first to see single-celled microorganisms, tiny plants and tiny animals using bacteria. Yet Leeuwenhoek never had the faintest suspicion that some of his animalcules caused humans to sicken and die. So this is a picture. In 1665, the English scientist Robert Hooke published a detailed drawing of a flea as seen through the newly invented microscope. London society clamored to see microscope images of things too fine for the human eye. And that's from a book of his pictures. That's more or less the state of knowledge in 1848. Few doctors have ever used a microscope because it is not considered a medical instrument. These microscopic animals might be marvels of nature, but no doctor suspects that they have anything to do with disease, let alone infections. Doctors in 1848 don't use the word infection, but they know its symptoms well. They call it sepsis, and they know from bitter experience how quickly a septic wound can go straight from slight redness to gross swelling to a fatal condition called gangrene. The doctors of 1848 don't realize that gangrene is the end result of bacterial infection. They don't realize that floating in the air on dust particles, lurking on fingertips, or growing on the shiny steel blades of their unwashed surgical scalpels are single-celled bacteria and other microscopic life forms. On the smallest surface, there are hundreds of millions of them. They represent thousands of species. They are tiny plants, tiny fungi, tiny viruses, and tiny animals. Among the microanimals are 2 particularly dangerous families of bacteria, Streptococci and Staphylococci, strep and staph for short. What doctors don't know in 1848, strep and staph do, that the broken head of Phineas is an ideal location to land. A wound is an open door. A cut or break in the skin lets staph and strep bacteria colonize the warm, wet, nutrient-rich cells inside. Once these bacteria get established in the body, they reproduce wildly. The body's immune system tries to kill the invading bacteria with an array of special immune cells, while bacteria try to protect themselves against immune cells by cranking out toxic chemicals. That's an infection. The site of this biological battle between the immune system and bacteria swells up and turns red. In 1848, science is still 20 years away from figuring out that infections are the work of living, that is, biotic things. It will take nearly a century for science to develop the first antibiotic, penicillin, to counter infections. In 1848, a young Frenchman named Louis Pasteur is studying chemistry in Paris. Eventually, Pasteur will unravel the 3 great biological mysteries of his time. fermentation, decay, and infection. All three processes are the work of living microorganisms. Pasteur will call them germs. Pasteur's germ theory will lead to a revolution in medicine. It will inspire an English surgeon named Joseph Lister to try and perform surgery in sterile conditions that exclude or kill all microorganisms. Lister will scrub his hands almost raw before operating. He will boil surgical clothing and instruments, and he will set up a machine to spray carbolic acid into the operating room to kill germs in midair. Lister's first sterile operations in 1868 will cut the number of deaths from infection after surgery by 90%. For the first time in history, doctors will help more patients with surgery than harm them with post-surgical infections.